Hello students, hope you are fine. So one more topic from endocrine system we are going to deal with today. So that is the reproductive hormones, right? And today we are going to discuss the male reproductive hormones. As we all know, right, that we always start with learning objectives. So learning objectives of today's lecture are uh, describe external and internal anatomy of testis. This is very much important because without a brief overview of anatomy, you will not get an idea what are the exact action of the hormone. Okay, so for that purpose, it is important. Okay, outline hormones of the male reproductive hormones and understand their physiological effects. And finally, will be an illustration of control of release of male reproductive hormones. Okay, so this is how we are going to uh, 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 cover today's topic. So there is three level management for hormone for endocrine gland, right, in human body, and definitely the reproductive hormones are not an exception to this. So the three level loop, the highest level management is the ultimate control is with hypothalamus. Okay, and hypothalamus releases this hormone. So this GN refers to gonadotrophin. Okay, gonadotrophin is actually a combined name of two hormones, that is FSH and ICSH, or you can also call this as an LH. Okay, these two together are referred as a gonadotrophin. So here hypothalamic hormone is a releasing hormone, right? Releasing means it stimulates the release of gonadotrophins, and that gonadotrophins are FSH and ICSH. Full form of FSH is follicle stimulating hormone, and ICSH, it is interstitial cell stimulating hormone. Okay, in female it is also referred as a luteinizing hormone. In male it is referred as a interstitial cell stimulating hormone. Right. So hypothalamus, wherever been receiving the stimuli, right, it will stimulate the release of gonadotrophin. Gonadotrophin, releasing hormone, wherever reaches to anterior pituitary gland, right, these hormones are released from anterior pituitary part, right, via hypophyseal portal circulation. And this portal circulation definitely is important because it does not allow the hormones to dilute in a larger quantity of blood, and also a prompt effect, right, on the anterior pituitary gland can be received. So once been released, these two will act on the gonads, right, and you know that gonad in case of male is testis. Okay, so this is plural, that's why I have mentioned as a TES. If it is singular, you will write it as a TES. Okay, so that is difference, right, because you will get alternate spelling in my presentation. So that case, you just remember that this is a plural. Okay, fine. So hormones of testis, yes, major hormone which you refer as an androgen, that is testosterone. Okay, and could be easily remembered because testis, right, a steroidal hormone that is released from testis, major one, that is testosterone. Additionally, you have two more important hormones, that is androgen binding protein and inhibin, right. We will discuss the function of each of this in detail, but this is just overview that what we are going to discuss today, right. This is how the control is achieved, right, for the release of these hormones from the testis, right, which is an primary sex organ in the male, right. So the question arises in your mind that what is a primary sex organ, and many of you falsely answer that penis is a primary sex organ. No, it is not. The definition of primary sex organ is, these are the organ, right, this is an organ which can form gamete. Okay, so the organ which is involved in gametogenesis is referred as a primary sex organ. So such a case, in case of female it is ovary, and in case of male it is testis, which is a primary sex organ, right? So this is a very important detail and a very basic information for all of us. Definitely, male gamete, which is produced by testis, is spermatozoa, right? So we commonly refer as a sperm cell. Okay, and there are millions of sperm cells been synthesized daily, right, after the puberty has been achieved, right? And this almost is lifelong, right, until the androgens are released in the male, right? After the puberty has been started, it is almost up to 70-80 years or maybe more than that also, that the spermatogenesis can happen inside the human body, right, in case of male. While in case of female gamete is definitely ovum, which is actually not synthesized by the ovary. Ovary synthesize only up to secondary oocyte, and if a fertilization process happens, then it may get converted to a ovum, right? So it is here we mention it as an ovum, but ovum actually does not get formed until there is fertilization. Okay, this is quite interesting to know, but here we are focused more towards the hormones, so that's why I'm not going in detail of oogenesis. Maybe later on we can have a healthy discussion on the process of oogenesis. Okay, so we have gone through the basic definition that what is primary sex organ, and this testis we are going to get in details of, right? So external and internal anatomy, right, that is histology of testis we are going to check. Now location is quite interesting and important, right, because in contrast to ovaries are present in abdominal cavities, while testes are present outside the abdominal cavities, and that is within a pouch which is called as a scrotum, right? You also call it a scrotal bag. So what is the reason that testes are located outside the abdominal cavity? While they are important because they are involved in spermatogenesis, and if they are so important, they should be protected and they should be within the abdominal cavity. But here the importance of this location is the temperature, right? So temperature is the reason why they are located outside the body. Okay, so temperature of testis is 2 to 2.5 degrees Celsius lower as compared to the internal body temperature. You know that internal body temperature is somewhere around 37.4 degrees Celsius, and in case of testis it is 2 to 2.5 degrees Celsius lower, right? Now definitely who will help in maintaining this lower temperature? It is definitely scrotum, right? But why the lower temperature is important here? What is the reason that you need a lower temperature within the testis? So answer is, to execute the process of spermatogenesis, it is very important to maintain lower temperature, right? At higher temperature, the process of spermatogenesis is not executed properly, right? So sperms are very delicate in nature. They are very prone to get damaged by temperature, stress, right? And that is why this location, right, is outside the abdomen. Shape and dimensions are not that important. Shape is oval, which you can just get an idea that length is somewhere around 2 to 3 centimeter and width is 4 to 5 centimeter, right? So from this dimension, that width is more as compared to length, you can just assume that shape has to be oval, right? So external anatomy is quite easy, and now we are moving towards internal anatomy. Now if you go through a medial longitudinal section of testis, you will find a structure like this, right? So one by one we are dealing with this. It is internally, it is covered by 3 coats, and that coats are tunica vaginalis, right? So tunica refers to sheath, that is cover, right? And vaginalis, that is again in Latin it terms the outermost, right? Vagina refers to outer, so that means it is an outermost a layer, right, in the testis. Middle one is a very important one, which is made up of a white fibrous dense connective tissue, and because it is of white color, that's why it is called as a albu, right? You know that wherever the word albu is there, say for example a protein, albumin. So a protein which is of white color, that's why we call it as albumin. So same here, we call it as albugenia. This is important because this tunica albugenia, it divides the testis into different lobules, right? So you can just see that it extends inward. And to form the septa. And this septa creates testicular lobules. We'll check later on that there are not less than 250 testicular lobules present in each testis. Okay, so structurally you have many units. This is one lobule, right? There are around 250 lobules, such lobules which are present in the testicular structure. Okay, the last layer that is present in the testis, right, covering layer, that is called as a tunica vasculosa, and that is the innermost structure, right, as you can see here. Okay, so these are the three layers. Now we are moving the details of the seminiferous tubule, right, which is present in these testicular lobules created by the tunica albuginea, right. So forgetting all other covers, focusing on the tunica albuginea. Now you can just see that now, because the septum created by this, now you have lots of lobules, and as we have discussed, it's approximately 250. Okay, and each of this you have each of these lobule you have presence of seminiferous tubules. Semini refers to seed, right? Ferrous refers to transfer or carry. So the tubes which carry the seed here, it means semen, or not semen but sperm, right? So a tube which carries the sperm, right, that is called as a seminiferous tubule. And functionally importance is it is the site for spermatogenesis, right? So spermatogenesis, if someone asks where does spermatogenesis happen, so answer will be seminiferous tubule, right? This is the reason why I have taken internal structure of testis in before discussing the effects of the hormone, because without you can say that details of seminiferous tubules, you will not get idea, right? What are Sertoli cells? What are Leydig cells? Right? What are spermatogonium? That idea you will only get if you get the internal structure, histology of seminiferous tubules, right? But before seminiferous tubule, we will complete the internal structure of testis, and then we will focus on this structure, seminiferous tubule. Okay. So considering the case that approximately there are 3 seminiferous tubules in a each lobule, right? So 250 multiplied by 3, approximately there are 750 seminiferous tubules in each testis, right? So quite huge number. Okay. Now this seminiferous tubules, you can just see that these are tubes, coiled tubes. These are seminiferous tubules, right? So spermatogenesis definitely happens here, but this whatever spermatogenesis that happens, it actually is divided into different process. One is the meiotic divisions, that your diploid genetic material, that is 2n, 2 sets of chromosomes, is now converted to haploid set, that is a single set, n, right? So the cells which are having 2 sets of chromosome, that is 2n, are now after meiotic division have haploid type of the chromosome, right? So only single set is there, but these cells have still not gained the functional capacity to fertilize the ovum, because the chromosomes are made half, but the structures like acrosome, right, which has to be made from Golgi apparatus, flagella or tail like structure, right, that is made up from the centriole, that all is remaining. And to attain that functional maturity, you need some time. That time is provided by these all structures which we are going to discuss now onwards. So until they are moving towards the urethra from this vas deferens will carry it to urethra. Before they reaches to vas deferens, they should attain functional maturity, and that functional maturity is provided by these all structures. Okay, so that is what we are going to discuss. Seminiferous tubules, as you can see that now they join to form a straight tubules. So here there are straight tubules, there are many straight tubules, you can just see that. And these straight tubules, now they create a network of tubules. So you know that reticule always refers to network, right? Even in first year anatomy, we have learned that reticulin is a protein present you can say that in connective tissue. So reticulin means a small network. Protein is making a small net like structure. That's why we call it a reticulin. So similar name you can think here, retetestis. So here it is a network. within the testis, right? So that is created, and that will allow the sperm to get some maturity. Okay, still this time is not enough, and in that case, after a few times, because spermatogenesis is continuous, so after a few days, this all sperm will now move to the epididymis. So this is the structure, epididymis, very long structure, coiled structure. So, but yes, definitely the process, right, or the structure which facilitates the movement of sperm from retetestis to epididymis is facilitated by this structure, which is very important male sex accessory duct, and that is called as a vasa efferentia. Okay, so again revising, seminiferous tubules. From seminiferous tubules, now there are straight tubules. These straight tubules connect the seminiferous tubules to form retetestis, right? So there is a network of tubules. This retetestis now will move its content in this structure, which is called as a vasa efferentia, right? Remember that vas deferens is a huge structure, right? Diameter is quite big. Here there is a small structure, and that's why we do not call it as a duct, we call it as a ductule, right? Additionally, because it moves the content outside, that's why also we call it as efferent, right? So vasa efferentia are these small structures, and there are at least 10 to 20 such tubules present, right, which are coming out of this retetestis, and they move their content to this part, coiled part, highly coiled part, which is called as epididymis. So epi refers to above, so here it is above, you can see that, and didymis refers to testis. So a structure which is above, right, which is above the testis, that is how the name has been arranged, right? That is how the etymology is for the epididymis. Now you will be surprised to know that this epididymis is a highly coiled structure, and considering that total dimensions of testis are 2 to 3 centimeter in length and 4 to 5 centimeter in width, the length of epididymis is somewhere around 6 meters. Yes, you heard it right. It is 6 meters. And this much long structure is important, is necessary, so that a functional maturation of sperm can be done. If epididymis is absent, this functional maturity cannot be gained, and then sperm will not have their ability to swim, right? So that is motility will not be because of that present in that case. Okay, so for that reason, epididymis is very important, right, structure. You can divide this epididymis in 3 different parts. This is called as a head of the epididymis. Latin you can call as a caput. So this is caput epididymis. This is called as a body part, right? So body, that is corpus, right? So corpus is referred as a body. So corpus epididymis, and this is called as a tail part, right? Tail is always referred as a cauda, right? C-A-U-D-A. So caput epididymis, corpus epididymis, and cauda epididymis. Between these two, that is body and the tail part, you have a duct of epididymis also, which normally does not allow the movement unless and until there is a relaxation, right? Which may be because of either excess of spermatogenesis that happens here, or it could be a sexual stimulation that will allow the movement of material. Right? Here also important that definitely sperms are motile, so they have flagella. So that is the case that they will be able to move. But second thing is you also need peristalsis movement, the movement that you learned in the digestive system. Such also a peristalsis movement is also observed in these structures, in epididymis as well as in vas deferens, and that is created by local hormones that are referred as a prostaglandins. Okay. Definitely once the epididymis releases content, now it will be taken towards the abdominal cavity by a structure which is called as a vas deferens, or it is called as a ductus deferens, right? So this is how the structure, internal structure of testis is arranged. Okay, this will not only help you in understanding the effects of hormone, but also you know that it's a part of your syllabus also in your second semester BPharm. So from that point of view, male reproductive system is very important, right? So their internal structure could be helpful. As such, you can just overall male reproductive system diagram is more asked, right? Female and male reproductive system, overall front view and the lateral view, but internal structure of testis we have gone through, right? So this is an overview, right? You can just see that these are seminiferous tubules which are present within the testicular lobules, straight tubules, retatestis, vasa efferentia. Then you have epididymis, which is here there is caput, that is head. Here there is body, that is corpus. Here there is tail, that is cauda. And then finally vas deferens. Vas deferens moves towards the abdominal cavity in a spermatic cord, right? And this spermatic cord via inguinal canal, it moves towards the abdomen. Fine. So this is the inguinal canal, right, by which the spermatic cord carries vas deferens towards the abdomen. Here over the this is a urinary bladder, right? So just let me give you over. This is abdominal cavity. This is urinary bladder. This is rectum, right? So here vas deferens loops over this urinary bladder, and then just beneath the urinary bladder, at the base of urinary bladder, it receives a duct from a reproductive gland, accessory reproductive gland, which is called as a seminal vesicle, where it is the major gland, right? There are three male sex accessory glands. One is seminal vesicle, second is prostate gland, right? This is only single. Seminal vesicles you observe two structures, right? So that is in pair, and one more structure is there which is called as a bulbourethral gland, small bulb like structure. You can just see that. So this is paired. Bulbourethral gland also is paired, and prostate is unpaired. Okay, again going back to the vas deferens. Vas deferens, it receives a duct from the seminal vesicle, and now it creates a structure here that is called as a ejaculatory duct, right? And this ejaculatory duct is under the control of autonomic nervous system, and that is stimulated wherever there is a sympathetic response. So wherever there is stimulation of sympathetic nervous system, then and then ejaculation could happen, right? Remember, from this space, this tube, only sperm cells have come, right? So here these sperms are received. Seminal plasma will be added by these three glands, right? So this, once it is mixed with this structure, now it will create semen, right? So semen is equal to sperm plus seminal plasma. What is seminal plasma? Seminal plasma is a fluid which nourishes, protects delicate structures of the sperm, right? You know that sperms have negligible cytoplasm, and they are not able to survive for longer duration without nutrition. So that nutrition is provided from the seminal vesicles. That is, they are rich in fructose, some of the vitamins, proteins, prostaglandins. Additionally, the secretions of all these three glands, they are alkaline in nature, which will help them to protect in the acidic environment of the vagina, right? So this all content of seminal plasma is very important to nourish and protect the sperm. So after this ejaculatory duct, now it is called as a semen, and this semen will passes through, right? Now you can just see that it receives this is the base of urinary bladder, and now you can call these two, once ejaculatory duct and a duct from the urinary bladder, now once they merge, now you call it as a urethra. In case of male, there is a common passage for urine and sperms and semen, and that's why this urethra is also referred as a urinogenital tract, right? Urinogenital tract means one which carries urine as well as the gametes. Both are carried, that's why you call it as a urinogenital tract. Okay. Urethra also can be termed on the basis from where it passes. Here it is a prostatic urethra. Here it is an intermediate urethra, because there is no such organ from which it passes. And then finally the urethra passes from the penis. That's why you call as a penile urethra. Or because this penis is having some sponge-like tissue, right, which are having sinuses that could be filled with blood, the process which happens in case of erection. So that is called as a erectile tissue. or a spongiosum tissue, corpora spongiosum and corpora cavernosa. So because of this name, this urethra is referred as a, you can say that, penile urethra because it passes through the penis, or it is also referred as a spongy urethra because it passes through the spongy tissue. Okay, spongy means they are having a structure, internal structure like sponge, okay, which can be filled with the blood. Whenever there is a filling of blood in this tissue, there will be enlargement of the penis, then there will be hardening or rigidity, and that is what is called as a erection, right? So erection is sexual stimulation, and wherever this sexual stimulation is maximum, this ejaculatory duct opens, which leads to release of this content. When the content are released from this ejaculatory duct, now it is determined that they will be released through this urethral meatus, that is an outer opening, right? So in the urethra. So this is external structure in case of male reproductive system. Exam point of view, this diagram also is important. You need to practice it for the exact drawing of this diagram. Okay. Moving back to the structure of seminiferous tubule, as we have seen that testis mainly is having the testicular lobules, 250 testicular lobules and almost 750 seminiferous tubules within these lobules, right? So each of these lobules are having a 2 to 3 seminiferous tubules. And what is the functional importance of these seminiferous tubules? Yes, it is the site for spermatogenesis. So for that purpose, how the spermatogenesis or which cells give rise to the sperm cells, you need to learn the histology, right? So each of these seminiferous tubules is lined on its inside, right? Remember, on its inside by two types of cell, and those cells are called as the male germ cells, or what we call as a spermatogonia, and second cells which are nursing cells which help in the nourishment of the spermatozoa, and those are called as a sertoli. Remember that the cell which we refer as Leydig cells or interstitial cells, they are present outside the seminiferous tubule, somewhere here. Okay, so that case you need to remember, right? You need to remember that Leydig cells are not present within the seminiferous tubules. Leydig cells are present outside the seminiferous tubules. Okay, so that is a very important, because sometimes we get confused with such, because of that such MCQs which may be asked in your examination. Okay, so this is the overall structure. Now, okay, fine. So this spermatogonia are type of a stem cells, right? Stem cells means immortal. They can divide, right, lifelong, right? So this is a layer, and they are arranged in a 4 to 8 layers, right? Once a meiotic division starts, right, you can just see that some of these cells will divide by mitosis. Initially there is a mitosis. You know that mitosis is the somatic cell division, and once this somatic cell division, right, now they, in presence of some of the factors, now they will be stimulated, right, to go for meiotic division, right? Meiosis, you know that, is the division by which the chromosomal material is reduced to half, right? And that is a special type of a cell division which happens in case of the germinal cells only, right? Yeah, this is very important, that germinal cells which are present either in male or female, both are always cube shaped, and that's why the type of tissue, if someone asks that what is the type of tissue present in case of the spermatogonia, right, or seminiferous tubule, so answer will be cuboidal germinal epithelium, right? So these are germinal cells, right? Once a meiotic division start, you can just see that variety of cells, right? That primary spermatocyte, secondary spermatocyte, then finally spermatid, and you can just see that spermatozoa, right? So this is after the meiosis process, right? And in between this, I have shown only one section, right? This is actually a circle. So just in middle of this seminiferous tubule, there is a lumen. Lumen means space, empty space, where these sperms will move to the outside towards the straight tubules and finally to the rete testis. Until they are ready to release, right? These sperm cells, they remain attached with these spatial cells. You can just see that. And these cells are none other than the Sertoli cells. So you can just see that if you want to differentiate, say for example, which of these cells are spermatogonium or a primary spermatocyte and which of these cells are Sertoli cells. So you can easily recognize the cells where the spermatids or spermatozoa are embedded. These cells are referred as a Sertoli cells, right? So their function is nourishment of the, you can say that sperms, right? They provide nutrition to the germ cells. In addition, we'll check more clearly in next diagram, but here you just have to understand they are the basis for the blood testis barrier. Now what is this? Now you know that in case of brain, right, because brain is very delicate structure, the toxins, right, which are easily able to travel through blood, they can enter the brain and they may damage the neurons. Such a case, such damage cannot be allowed by our body, and that's why your neuroglia that is present in the brain along with the blood vessel, they have a very tight junctions present within these two lining, and in that case they do not allow the easy diffusion of such water soluble molecule. The molecule has to cross the cell membrane, and because cell membrane is a selectively permeable, that's why it will not allow an easy entry of such toxins. That is what a blood brain barrier is. Similar type of barrier you can consider for the testis. So toxins, their entry is prevented from the blood into the testis by this barrier, where the sustaincular cells, that is your Sertoli cells, are having such tight junctions which will not allow the water soluble molecules to enter inside the cell. Okay. Additionally, Sertoli cells, they are having endocrine function also, which is of our importance of today's topic. So they produces hormones, and there are two hormones that they are able to produce. One is inhibin, right, and second is androgen binding protein. Additionally, wherever, because spermatogenesis, you have large quantity of, right, millions of sperms being synthesized, right, in each ejaculation you must be knowing that there is the volume of semen is somewhere around 3 to 4 ml, and each ml of semen contains 20 to 120 million of sperms. So on an average, right, during each ejaculation, not less than 300 to 400 million of sperms are released, and this many sperms are synthesized. So in case it is possible that some of these sperms may get damaged, right, in the process of their formation only, or sometimes they may get dead also, and such dead sperm cells has to be removed by the process of phagocytosis, and that process is also done by Sertoli cells. So multifunctional importance you can consider, right, but major function is definitely this, that they provide nutrition to the germ cells. Okay. Third cells are very important, which are present outside. Inside there are only two cells, and outside the seminiferous tubules, that is within the interstitial space. Interstitial space means space between two tissue, right, two seminiferous tubule. And from the scientist who just discovered these cells, we need to give credit to them, to him, and that's why they are also called as a Leydig cell, right? So interstitial cells or Leydig cells, they are located outside the seminiferous tubule, that is in the interstitial spaces. So here now it is very easy to understand. This is a spermatogonium, right? One is called as a spermatogonium, many spermatogonia, right? Few of this, under the influence of testosterone, this will be stimulated to go under the meiotic division, and you can just see that successive meiotic division can eventually lead to the haploid cell. This haploid cell by a process, spatial process, right, by which cell changes its morphological characteristic. It moves to a specialized cell, that is a sperm cell. That is called as spermiogenesis. And this, until this is capable of swimming, it is, you can just see that it is remaining attached to this large cell, and these cells are nothing but they are Sertoli cells. Okay, so Sertoli cell, now it is very easy. These are blood vessels, right? These are blood vessels. Some of the immunological cells, blood vessels, they are also present, that you can just observe in this section. And this large cell, these are interstitial cells or Leydig cells, which secretes the testosterone. Okay, so endocrine point of view, two cells you need to remember: Sertoli cells and this that testosterone. Okay, so effector cells are definitely this spermatogonium, and then primary spermatocyte, secondary spermatocyte. But the cells which release these hormones are this. Okay, you'll get an idea when I will go to the next slide. This is an scanning electron microscope diagram, which again shows the seminiferous tubules, and you just see that this is the protective layer, connective tissue layer, spermatogonium. Just see that 4 to 8 layers, right, of these cells which are lining these seminiferous tubules from its inside, and some of these cells are Sertoli cells. It's difficult to observe from this diagram. And these cells, right, which are having flagella, these are nothing but they are sperm. Okay, so they are emptied in this lumen, right? This is a space, it's a lumen. So here is where they are released. Okay, so this is the diagram. Okay, I hope it is now very clear to you. Done. So these cells, what is the function? They release a very important hormone, and that hormone is testosterone. Okay, so that's a major androgen. And now we are moving towards the reproductive hormones in case of male. Now remember that a hypothalamic hormone is GnRH, and GnRH is a gonadotropin releasing hormone. This releasing hormone will stimulate adenohypophysis, part of adenohypophysis, that is anterior pituitary. Anterior pituitary will lead to release of, right, gonadotropes will release FSH, which is follicle stimulating hormone, and LH, or in male it is referred as a ICH. Now it is very clear to you, right, that what should be their full form. Okay, so who secretes this FSH? Remember, yes, it is pituitary gland. Which part of pituitary gland? It is anterior pituitary, or you can also call it as an adenohypophysis. But if someone asks that where do they act, so what are the effector cells for FSH, then answer will be Sertoli cells. Remember that in very first lecture of endocrine system, that we have learned that hormones are released in the blood circulation. It does mean that, like, endo means within, crine means release, and without duct, these chemicals are released directly into the bloodstream. So hormones will go everywhere in the body, but they will act only on the cells which are having receptors, right? So wherever FSH receptors are present, that cells are effector cells for this hormone, and those receptors are present in Sertoli cells. Okay, so once been released, they will act on the FSH receptor. Again, if someone asks you what will be the location of FSH receptor, whether it will be on the cell membrane or whether it will be within the cytoplasm or nucleus, so your answer will be on the cell membrane, because FSH and ICSH both are peptide hormones. Okay, and you know that proteins, peptides are water soluble, they cannot cross the cell membrane, so they will act on the cell membrane bound receptor. Fine. Sure. So what will be the effect? Once been acted, once been bound to the receptor, this will cause the cells to grow, right? The Sertoli cells will grow, and they will secrete various spermatogenesis substance. And also important that they stimulates, FSH stimulates the release of androgen binding protein. Without androgen binding protein, the concentration of testosterone within the seminiferous tubule is not possible, right? So that's why the importance of ABP is there, and if ABP is important, definitely FSH is important. Okay, and that's why we say that before puberty, you don't have the process of spermatogenesis. You don't have development of primary and secondary sexual characteristic being developed. Why? Because anterior pituitary gland is not releasing FSH, is not releasing ICSH. Without FSH, Sertoli cells cannot function, right? Without ICSH cells, Leydig cells cannot function. Okay. Second cell, second hormone, that is ICSH. Again, from where it is secreted? Yes, definitely it has to be pituitary gland. Which part of pituitary gland? Again, answer is same, is anterior pituitary or adenohypophysis, right? From the cells which you call as a gonad or trough, right? So this is the secretion. Effector cells, what just say for example, here we have remembered that effector cell in case of FSH, right? These are follicular cells. So that's why follicle stimulating hormone or Sertoli cells. Similarly, here in this case, ICSH, what will be the effector cell? Effector cell will be interstitial cell or Leydig cell. Okay. So it stimulates the Leydig cell and function is to secrete the testosterone. Right? Remember, testosterone is a steroidal hormone, and because it is steroid in nature, it will act on the cells and the receptors which are located within the cell, right? And that is on the nucleus cell. We call this receptor as a nuclear receptor. Okay. So this is an anabolic hormone. It's a lipid soluble hormone and easily able to cross the cell membrane, thus acting on the nuclear receptor and activating the anabolic process. All changes which are related to the primary and secondary sexual characteristic in male, they are due to the release of testosterone. Right? We'll check the effects, physiological effects in the next slides. Yes, these two hormones are still remaining, and that is androgen binding protein and inhibin. Okay, actually three remaining. You need to also discuss details of testosterone, right? So first two hormones which we have learned, that is FSH and LH, are from the pituitary gland. Now these two hormones are from the Sertoli cell, right? So this is the one we have learned that FSH, once been bound to the receptor, will stimulate Sertoli cell to release ABP, and this ABP it is helping in the concentration of testosterone inside the seminiferous tubule. Right? So how does it concentrate the testosterone? Let's have a look. You can just see that this is seminiferous tubule, this is Sertoli cell, and this is where your FSH is acting. So FSH is acting on the membrane bound receptor, and then it stimulates the release of ABP, androgen binding protein, and you can just see that once testosterone is entering, because it is lipid soluble, it will be able to cross cell membrane, but then it has to be concentrated within this cell. So in that case, this androgen binding protein will help in the concentration of this testosterone, and you can just see that process of spermatogenesis also will be facilitated. Right. So this is regarding ABP. Next is inhibin. Now, as the name suggests, right, you must be aware of wherever there is a word IN. Right. Say for example, albumin. So what does that IN refers to? IN refers to protein, peptide. So it's a peptide hormone. Okay. A peptide hormone which inhibits what it inhibits? It inhibits spermatogenesis. That's why you call it as a inhibin. Right. Now you must be knowing, or you must be guessing that why do you need inhibition of spermatogenesis? Everything has to be in balance, right? Anything in excess is poison, you know that. And in that case, process of spermatogenesis also has to be balanced. It has to be stimulated by testosterone and androgen binding protein. But simultaneously, wherever there is an excess of spermatogenesis, it has to be stopped. And that regulation is done by inhibin. Again, it is secreted by Sertoli cells only, but it's a checkpoint, right? Wherever there is an excessive spermatogenesis, so in response to that, the Sertoli cell will release inhibin. What was the controlling factor in case of ABP? It was a release of FSH. When FSH is released, then ABP gets released. What is the controlling factor in case of inhibin? Wherever there is an excessive spermatogenesis, then the release of inhibin will be stimulated. Okay, so it's a negative feedback message, right? You know that there are two types of loop existing, right, for the control of the hormones. One is a negative feedback loop. Second is a positive feedback loop. Positive feedback loop rarely happens in our body, and few of those examples, only 2 examples of that include release of oxytocin during the process of parturition, that is childbirth, and second is estrogen as such, right? If it is released alone without progesterone, then estrogen do stimulate the release of FSH. These are 2 examples where positive feedback loop happens. Otherwise, in all hormonal release, always there is a negative feedback loop. And a classical example that I always give to you, that suppose you have appetite, right? Bhook lagi hai, and then you are sitting, right? Your mother, right, definitely will give you food. Maybe you are starting eating chapati. So 1, 2, 3, 4, maybe 5, right? And then after your stomach will say no, nahi bhai have joiti nathi, have bhook lagi nathi, my abdomen is now full. So that is what, now your stomach has given a message to brain that no, now I don't need to eat. And that is what you can consider a case in case of negative feedback loop that is happening in case of by these Sertoli cells through release of inhibin, right, to the anterior pituitary and hypothalamus, right, which results in the suppression of synthesis and release of FSH. Okay, so this is regarding inhibin, right? So the function is to suppress this spermatogenesis. Okay, last one and the major one, most important one, that is testosterone. I need answer from you. You can pause the video and you can give the answer. Who secretes the testosterone? Yes, it is interstitial cells or Leydig cells. What is the function? So it is important, it is essential for spermatogenesis to happen. Meiosis, the process of the spermatogenesis only happens if there is presence of testosterone. Without that, it cannot happen. So for sperm production, it is necessary, it is mandatory. Okay. Second one is development of secondary sexual characteristic. Right. So apart from the development of the physiological changes in the testis, which are related to spermatogenesis, this is what you call as a primary sexual changes, right? But then definitely there will also be many symptoms, many signs which you can consider as a development of a secondary sexual characters. Few of these examples we'll discuss. Say for example, development of beard, mustache, right, daddy mooch. Okay, then you can say that the lowering of pitch in case of voice, right, that is we call as a hoarseness of voice, right, thodo awaj ghero thai jay. That happens in case of puberty. Why? Because the mucosal folds which are present in the larynx, they are influenced by the presence of testosterone. Testosterone in female is less, that's why they have a high pitch, right, awaj thodo tino hoy. In case of male, there is low pitch, so that is leading to the hoarseness of voice. Similarly, the skeletal muscles develops more in case of male, so there's muscular structure, right? The male body is designed to perform hard work, so that muscularity of the structure, right, of the skeletal muscle, definitely it is under influence of testosterone. Erythropoiesis also is stimulated more in presence of testosterone, and that's why the values of RBCs, right, in case of male are higher, values of hemoglobin in case of male are higher as compared to female. Okay, so all of this you can consider as an, right. Also not to forget development of hair in chest region, right, in pubic region, that also you can consider as a secondary sexual characteristic, right. Definitely in addition to FSH, testosterone also stimulates androgen binding protein secretion. So it's a vice versa situation. Androgen binding protein helps concentration of testosterone, and testosterone will further help the stimulation of androgen binding protein, right. So these two will, you can say that, have a mutual sort of an phenomena by which they will increase their action. Finally, wherever this testosterone is released in more quantity, excessive quantity itself, it will act as an auto-inhibitory mechanism. It will give a negative feedback mechanism, right. So there will be suppression of gonadotropin releasing hormone from hypothalamus, and further there will be suppression of FSH as well as LH or ICSH, both. from the anterior pituitary gland. So it gives a negative feedback mechanism, right, to both of this hypothalamus and anterior pituitary. Now, last function is a very important that happens in case of intrauterine life. Intrauterine life means still the fetus is the case, or it is the structure is present within the womb, within the uterus. Now, in last trimester of this intrauterine life, there is a release of a very low quantity of testosterone, which is as low as 30 nanogram per ml. And this quantity, right, if it is released, this will help movement of testis from abdominal cavity towards the scrotum, right, that is outside the abdominal cavity. And this process is called as a descentation. Descentation means movement of testis from abdomen towards the scrotal bag. This is important because if this does not happen, it will lead to a condition which we'll discuss in next slide. But then the testis will not be able to move outside, right? So this concentration of testosterone is very important for the embryonic development process, right, which we call as a descentation of the testis. If this process fails, right, say for example, if this descentation does not happen, then this testis will not move outside, and that we call as a undescended testis. Okay, undescended means the descentation has not been done. Or in that case, you can also call crypto. Crypto refers to hidden. Testes are not been visible from outside. That's why crypto. And orchid refers to testes again. Ism means a process. So process where the testes are hidden. That's why you call it as a cryptorchidism. So cryptorchidism is a process when testes do not descend because the testosterone levels in the intrauterine life are not enough to push them in the scrotal bags. Such a case, there will be two problems. One, we have referred that without scrotal bag, the spermatogenesis will not execute properly. That means sperms are so delicate that at higher temperature spermatogenesis is not easily facilitated, right? And second, if they remain in abdominal cavity, there will be a cancerous tissue, there will be development of malignancy, right? So functional and anatomical development cannot happen. That is one thing. Second thing is malignancy, and for that reason a very simple surgery is been done, which is referred as a orchiopexy. Orchiopexy is a surgery, right, in child, right, where there is a surgical fixation of testis, right. Doctors, right, surgeon, definitely they will move the testis from abdominal cavity in the scrotal bag by surgery. So pexy refers to surgical fixation, and orchio, as we have seen here, it refers to testis. That's why we call this as a orchiopexy. Okay, so these are some of the terminologies which are related with the development of testis, and that also simultaneously we have learned. One more process which definitely will not happen in case of the human, but in case of animals this has been done. It's called as an ectomy. Ectomy refers to surgical removal, and gonad refers to definitely the primary sex organ, that is testis. And surgical removal of gonads is called as a castration. If it is a surgical removal of testis, is called as a castration. That is done in animals, right? Say for example, bulls are converted into bullocks, right? So, and the reason is because if you remove testis, testosterone values will be lower. If testosterone is less, the aggressive response in male will also be less. Okay, and so them to make more obedient, so that they will obey whatever their the person who are taking care of that animals, right? So there it will help to make it more obedient, right? Because it will decrease the testosterone. Definitely in behavioral changes as compared to female, males are more aggressive, and the reason again is testosterone. Okay, so castration is a very simple process. Yeah, one more term from this suffix I would like to mention you. You just go through a term which is called as a vasectomy. What do you mean by vasectomy? Okay, fine. Consider this as an assignment. What is vasectomy? Okay, that you need to find. Second question is, if there is a vasectomy, will that person, will that male will be able to ejaculate or not. Ejaculation process will be done in the person who has undergone vasectomy, right? That is what you have to find out. Okay, so definitely if you don't find, if you are not able to find, you can ask me in my WhatsApp, right? So I will give you details regarding that. And you know that vasectomy is a very important process for which is a very important method for family planning, right? Sure. This is an overall summary of today's lecture. You know that hypothalamus releases gonadotropin releasing hormone. So this RH through a portal circulation, hypophyseal portal circulation, will go to anterior pituitary, where they will stimulate the release of ICSH, right, interstitial cell stimulating hormone, and FSH, follicle stimulating. This further will go to their target organ, endocrine target, that is testis, and there they will act to stimulate two different cells, right? That is Leydig cells. You know that ICSH act on the Leydig cell, which will stimulate Leydig cell to release testosterone. Testosterone is responsible for development of primary and secondary sexual characteristic. Wherever been released in excess, it will give an auto-inhibitory mechanism, excess level. So there is a negative feedback loop, and this will stop release of ICSH and GnRH from the hypothalamus. Similarly, FSH acts on the Sertoli cells. Sertoli cell releases two hormones. One is androgen binding protein. This helps in the spermatogenesis by concentrating the testosterone in the seminiferous tubules. Okay. And lastly, wherever there is an excessive spermatogenesis, then it stimulates the release of inhibin from Sertoli cell, which will give a negative feedback message to anterior pituitary and hypothalamus. This is the best way by which I can summarize the action of all male reproductive hormones, right? So with this I have completed my lecture. Thank you very much. If you have any doubts regarding this lecture, this topic, you can definitely ask on my WhatsApp number. Thank you very much.